molecules that are polar, right? That are water soluble. So polar, large, water soluble molecules will not be able to cross the cell membrane. Why? Because of the fact that phospholipids have a hydrophobic interior. Since the interior of the phospholipids, the inner side of the cell membrane is hydrophobic, so polar, large, water-soluble molecules cannot cross through the cell membrane. Only small, non-polar molecules like oxygen and carbon dioxide will cross through. Soluble molecules, water-soluble molecules, or large molecules will need to travel across the cell membrane with the help of some protein channels. We had discussed in the last class our transport proteins. So transport proteins, there are different types of them. We will discuss them in a bit. So transport proteins are going to help in these substances going from one side of the cell membrane to the other, right? So phospholipids, by being hydrophobic, by having hydrophobic tails, they make sure that our molecules are not easily able to diffuse in and out of the cell, right? Only oxygen, carbon dioxide, other small non-polar molecules can diffuse through, but our molecules that are really important, like glucose, it can't just diffuse in and out. It will only diffuse in and out if the cell needs it, right? It's regulated, it's controlled. So that's one of the features that we have studied about phospholipids. So it's stuff that is inside of the cell, the proteins that are inside of the cell, the lipids that are inside of the cell, the carbohydrates inside of the cell. They can't just move out of the cell because the cell membrane will not let them go out. Along with that, if you had remember, I told you that some phospholipids as signaling molecules, and signaling molecules is also a topic that we will take a look at. In this chapter, we will study about how does the cell signal each other, right? Now, after that, if you remember, we had of all the structures we had talked about in the cell, so we had phospholipids, we had proteins, and we had cholesterol. Let's talk about cholesterol. If you remember, I had mentioned how a cholesterol works. What had I told you guys? I had told you guys, especially about the cholesterol, I had specifically mentioned that cholesterol helps in regulating or controlling the fluidity of the cell membrane, right? The cholesterol helps in regulating and in controlling the fluidity of the cell membrane. So the cholesterol will make it so that when the temperature is too low, the cell membrane doesn't become too stiff, too rigid. And when the temperature is too high, the cell membrane doesn't become too fluid, right? So how does it do that? When we talk about it, just like phospholipids, right? Just like phospholipid, cholesterol has a hydrophilic head and hydrophobic tails, right? They have a hydrophilic head and they have a hydrophobic tails. So they fit neatly between the phospholipid molecules. So they fit fine. They fit easily between the phospholipid molecules. They fit very easily between the phospholipid molecules. Now, when we talk about cholesterol, so cholesterol is mostly found in animal cells, by the way. We don't find a lot of cholesterol in plant cells, and prokaryotes especially do not have any cholesterol at all. So it's a feature that's mostly seen in animal cells. Now, when we talk about it, how does cholesterol work? What is this cholesterol doing that is allowing it to control how a cell membrane is going to be fluid, or how stiff or rigid the cell membrane will be. How does it control it? So it controls it in a certain manner. At low temperatures, since the temperature is low, so molecules have less kinetic energy, right? So what happens is that the cell membrane can become rigid. So cholesterol keeps the cell membrane fluid and prevents it from becoming rigid. How does it do that? It actually prevents the phospholipid tails from packing close to each other. So think of it this way, that it's a really low temperature. The cell is at a very low temperature. Now, since the molecules have less kinetic energy, the phospholipid tails will not be moving around that much, right? Since they won't be moving around that much, they will be still. So there is a chance that, they will start to compact together. They will start to pack against each other. They will start to come against each other. And if they pack against each other, if they, sort of come together with each other, the cell membrane is going to become stiff because the tails are not moving anymore. The cholesterol molecule is sort of like an interference or a barrier. Since there are cholesterol molecules present between the phospholipids, they won't let these tails become compact. They won't let these tails stick together. They won't let these tails pack together, right? So what happens that since the tails are not packing together, they stay separate from each other. The cell membrane remains fluid, right? And this is what it actually looks like. We have this is our phospholipids, for example, right? And we have our phospholipids. Now, what is going to happen that, generally speaking, normally what would happen is this. Our phospholipid tails would come together. They would pack together with each other, right? They would come together, they would pack together like this. So they would come together, they would pack together, they would become all compact, right? A cholesterol molecule in the middle changes the story. How does a cholesterol molecule in the middle change the story? A cholesterol molecule in the middle will not let these tails become compact. It won't let these tails pack together. So the tails will remain separate from each other. Hence, our cell membrane will maintain its fluidity. The cell membrane is going to maintain its fluidity. It's not going to become too compact. So that is how cholesterol works at low temperatures. Okay. So what happens that it prevents the phospholipid tails from packing together, and hence maintains maintains the fluidity. It also does one more thing. The interaction of the phospholipids with the cholesterol molecules also helps to stabilize the cell at a higher temperature. Because what would happen at higher temperature? At a higher temperature, there is a risk that it's going to become too fluid. That's the problem. Too much kinetic energy. They've become too fluid, right? So these molecules, cholesterol molecules at high temperature, they will interact with the phospholipids. And when they will interact with the phospholipids, they will make sure that the phospholipids stay stable. Even with low temperature, they stay stable and maintain the fluidity of the membrane. Make sure that the membrane doesn't become overly fluid, right? So this is the usage of cholesterol. Other than that, what else does cholesterol do for us? Cholesterol also maintains the mechanical stability of the membrane. Without cholesterol, the membranes would quickly burst. If there was no cholesterol to keep the membrane stable, they would quickly burst or they would quickly break apart. What else? How else is cholesterol helping us out? So another way that cholesterol helps us out is that the hydrophobic region of the cholesterol molecule helps prevent large ions, large polar molecules or ions from passing through the membrane. So it helped out the phospholipids. The phospholipids were preventing the non-polar large water-soluble molecules from crossing through, right? So the cholesterol helps in that. It also helps in controlling who is coming out of the cell and who is going out of the cell. So that was the different role of phospholipids. This was the different role of cholesterol within the cell. Now, if we are talking about our structures within the cell, there is another important structure within the cell membrane that's really important, right? We are going, when we talk about, we're going to be talking about what? Proteins. Proteins is an extremely important, you can say, component of the cell membrane, without which our cell membrane will not be able to work the way it is. When I say proteins, so let's go ahead and talk. Now, if you remember, I had mentioned that we have glycolipids. Glycolipids are lipids with a small carbohydrate chain. And we can have glycoproteins, which are proteins with a small carbohydrate chain. And then we can have proteins as it is. We can have proteins directly, right? Now, what do we need to know about proteins? Now, when we talk about glycolipids and glycoproteins, so the carbohydrate chains that we have, these carbohydrate chains basically project like an antenna. Like we have antennas, I'll draw it for you so you can see. They project like antenna into the tissue fluid or the fluid surrounding the cell, right? They form hydrogen bond with the water molecules and help stabilize the membrane structure. The carbohydrate chains that I'm talking about, these carbohydrate chains, they form a sugary coating around the cell, and this sugary coating is called a glycocalyx. So it looks like this. If I go ahead and draw the structure of the cell membrane for you. So when I talk about the structure of the cell membrane, as you know quite well, we have phospholipids, right? Along with phospholipids, we have proteins. If you remember, we had studied two types of proteins. Those proteins that go throughout the entire cell membrane. We call them intrinsic proteins or transmembrane proteins. Then we can have certain kind of proteins that are known as extrinsic proteins, right? So we had talked about our extrinsic proteins, those that are found on the outer side or the outer layer, the hydrophilic layer of the cell. And we had talked about the fact that we can have, so these are the different types that we had discussed, right? Along with that, we had talked about how we can have, cholesterol molecules within the middle, and we had discussed how this gives us a fluid mosaic appearance. Now, when we talk about our proteins, so I have my protein, right? Now, if on top of this protein, I have now, if on top of this protein, I have, let's say, a carbohydrate chain. So this carbohydrate chain, can you see how it's coming out like an antenna? It literally looks like there is an antenna attached to my protein. Or if I have a lipid, a phospholipid, and on that phospholipid, I have a carbohydrate chain attached. So that carbohydrate chain looks like an antenna. And this carbohydrate chain, what does it do? It actually, what it's going to be doing is that it is actually going to be helping in stabilization of the cell membrane. And how is it helping in that stabilization? By making hydrogen bonds with the water molecules, right? By making hydrogen bonds with the water molecules. So by that is one of the functions. I'm not saying that's all of the functions. We are going to be taking a look at a few interesting functions in a bit, but that is one of the functions that we have with our um when we talk about it, our glycoproteins and glycolipids. Glycoproteins and glycolipids have other functions as well. So they're going to be involved in making hydrogen bonds, which is going to help in stabilizing our structure. And this is going to go ahead, and this particular antenna is going to be acting as our glycocalyx. Now, continuing on from where we were talking about, so that is one of the function of these glycolipids, that is one of the function of the glycoproteins. Now, in animal cells, the glycocalyx is mostly made up of protein. Okay. In animal cells, the glycocalyx is mostly glycoprotein. But the situation becomes slightly different when we talk about your plant cells. In plant cells, it is mostly glycolipids that make the glycocalyx. Now, another function of these molecules just give me 1 second. Another important function of these glycoproteins and glycolipids is that they act as receptor molecules. What do we mean by that? So they basically, when we go ahead and talk about it, they are going to, so you must have heard multiple times that, oh, an hormone came and it binded to the receptor, or a chemical came and it binded to the receptor, right? So these antennas, these glycoproteins, these glycolipids also act as receptor molecules. Now, we have three classes of receptor molecule, or three groups of receptor molecule, right? The three groups are like this. We can have, for example, signaling molecules. And we will study cell signaling in a bit, so it will become a bit more clearer what I'm talking about. Signaling molecules are molecules that help coordinate the activities of the cell. How exactly are they going to help coordinate the activities of the cell? So these receptors are going to recognize your molecules, like you have your hormones, right? They recognize signaling molecules specifically like hormones. So for example, if you have a growth hormone, the growth hormone tells the cell that it needs to start making more protein, for example, so the person can grow. That is a signaling molecule. It's giving the cell a certain signal, right? So we can have signaling molecules or we can have neurotransmitters. What are neurotransmitters? Neurotransmitters are compounds, are chemical compounds that help in the conduction of nerve impulses. So for my nerve impulses to be conducted, for my nerve impulses to pass on from one nerve cell to the other, I need these chemical compounds known as neurotransmitters, right? Now, what is going to happen when a signaling molecule binds to the receptor? What happens? A series of chemical reactions is triggered inside the cell. Okay. So what is going to happen? That is going to be signaled inside the cell. And then the body or the cell is going to respond in the way that it is supposed to. For example, if the body is producing glucagon, remember glucagon we studied when we did metabolism or your glucose regulation in IGCSE. So if we have, for example, a glucagon will bind to the receptor molecules on liver cells, specifically liver cells. Only liver cells will have these signaling molecules. And what's going to happen that it's going to, when it's going to bind to the molecules, a series of reactions will start inside the cell. And the ultimate result of those series of reactions would be that the glycogen, the stored form of glucose, the glycogen will be broken down to release glucose, right? So that is how signaling molecule works. And we will study the signaling process in a bit more detail. Then they can act as receptors for endocytosis. Endocytosis is also a topic we'll study in this chapter. But what actually is endocytosis? Endocytosis is that when a cell takes in a substance. When a cell takes in a substance or it engulfs a substance. Okay. So, for example, you guys must have studied this with phagocytes. Remember, phagocytes would engulf the bacteria. So, cells don't just do it for bacteria. They can do it for food molecules. They can do it for other things as well. That's endocytosis. So, for endocytosis to occur, again, we need receptors, right? So, what happens that these receptors, these glycoproteins, these glycolipids, they will bind to the molecules that are parts of the structure to be engulfed by the cell membrane. How does it do that? To be a bit more explanatory, it works in such a way that you have, let's say you have a white blood cell. That white blood cell wants to eat the bacteria. So the glycoproteins and the glycolipids on top of the cell membrane of the phagocyte, they will bind to the cell membrane or cell wall of the bacteria. And when they will bind to the cell wall of the bacteria, that will allow the cell to actually eat the bacteria and engulf the bacteria, right? So for that, we need these particular structures. We need these glycoproteins and these glycolipids. The glycoproteins and the glycolipids are the ones that are going to go ahead and actually grab these molecules, grab these bacteria, and actually go ahead and allow the cell to do the engulfing. After that, the third group of receptors is something we're not going to be talking about, but they help in cell adhesion. What is cell adhesion? So you must have seen that your cells are often adhered to each other, right? The cells are often attached to each other. And because the cells are attached to each other, that's why we have, layers of cells. That's why we get our tissue. That's how we get organs. So these proteins, glycolipids and glycoproteins also help in that. They also help the cells attach to each other so they can make tissues, they can make. Now, so these are some of the functions that we have. So they can act as receptor molecules. So they can act as glycocalyx. That's the first function related to them, that they act as glycocalyx. That's their first function. The second function that they have is that they can act as receptor molecules, and we have talked that there can be 3 types of receptor molecules. The third function that we are often going to go ahead and see with these structures is that they are going to be acting as transport proteins. This is a point that I mentioned previously as well, that they can go ahead and they can act as transport proteins. Now, when we talk about transport proteins can be of different types, right? We can have either channel proteins. So channel proteins are proteins that are going to have a simple passageway through them, and the molecules can diffuse in and out of the cell through that passage. Or we can go ahead and we can have carrier proteins, right? Carrier proteins are proteins that are actually going to go ahead and help in, for example, I'll give an example, active transport. So carrier proteins will help in active transport. We are going to study in this chapter about facilitated diffusion. So you will see over there how these carrier proteins are going to be helping us. These proteins can also act as gated channels. What are gated channels? So gated channels, again, they're channels. They're like a pathway, but they are gated, meaning that if a specific molecule will come and bind, only then they will open, right? And we will study all 3 of these in more detail a little bit on when we talk about diffusion, facilitated diffusion, active transport, and all that. I will explain to you guys what these 3 types of proteins are and how do they work. Along with that, what else do we see? So we also go ahead and see them acting as antigens. What are antigens? So antigens are, specifically speaking, think of them as like the identification marker of your cell. The cells of our body have a very specific antigen. And our immune system recognizes that antigen. So this is how our immune cell is able to tell whether the cell is from our body or whether it's coming from the outside, right? How exactly is it able to do that? How exactly is the body able to recognize if a particular cell is from the body or is it something foreign? Is it something from the outside? So it looks at antigens. Antigens are like the ID card of the cells. The immune system will look at the antigens and it will recognize that, oh, okay, this antigen is from our body. This is our cell. If a bacteria is going to come from the outside, something comes from the outside, doesn't have to be bacteria, anything comes from the outside, the body will recognize that, oh, this is not our antigen. So the antigens, what are they? Our glycoproteins and glycolipids. The glycoproteins and the glycolipids act as antigens. They are the antigens, right? And they help the immune system in recognition by the immune system. We call it cell-to-cell recognition, that the cell can recognize each other. And this cell-to-cell recognition is very important. Why is it so important? Because if the immune system is going to be able to differentiate between our cells and the harmful cells, it's going to be able to defend us, right? It's only going to be able to defend us if it can recognize between our cells and the cells from the outside. And that is where a problem can occur. There are certain diseases which we call autoimmune diseases. What are autoimmune diseases? Autoimmune diseases are diseases where the immune system of our body will attack us. My own immune system will be destroying my own cell. And this is the reason for it, because there is some kind of problem in the immune system, because the white blood cells, there is some kind of problem going on with them. They are not able to recognize our antigens. So they will not recognize the glycoproteins and the glycolipids on my cell. So they will think my own body cell are from the outside and they will start attacking, right? So this is really important that we have these antigens that will allow us to differentiate between them, right? And different cells are going to have different antigens. So you know how in your blood groups, we have got blood group A, blood group B, blood group O. Now you can understand what the difference is. The difference is that blood group A will have antigen type A, the type A antigen. Blood group B will have type B, right? So the antigens will be different. That's why we have different blood groups. And that's why if I accidentally transfuse the wrong blood group in a person, that person will get a reaction because the body will not recognize the antigen, right? It can also go ahead and act as your enzymes. That is another use for your proteins, for your glycoproteins and glycolipids, right? We can have enzymes. And these enzymes, if I were to give you an example are enzymes found in the cell membrane of of the surface of small intestine, right? In the small intestine, we can have these enzymes, and these enzymes are actually in the cell membrane. So the surface of the small intestine, the lining of the small intestine, the cells of that surface, they will be attached to the cell membrane, these enzymes, right? And they help in breaking down food molecules. So these are the different uses that you are going to have related to your proteins. Any confusion so far? Does anybody have any question? Would anybody like to ask anything?
No.
Arush, I hope everything clear. Okay, if nobody has any questions, let's go ahead and take a look at a super important topic that we have to go over, and that is cell signaling. Wow, what is cell signaling, right? Cell signaling is when the cells are basically sending messages to each other, right? They can send messages such as divide, start cell division, start cell growth, start producing this hormone, start producing this chemical, start breaking down glycogen. That's the one we discussed, right? So for cells to coordinate with each other, right? For cells to coordinate with each other, for cells to work together, we need cells to be able to communicate with each other, right? And that is where cell signaling actually comes in. So, cell signaling is actually done when you go ahead and talk about it. Cell signaling is actually done with the help of signaling molecules. Now, for cell signaling to occur, I need two things. I need to have my receptors on the cell membrane, which we discussed, that the glycoproteins and your glycolipids can act as signaling receptors on the cell membrane. After that, I need small signaling molecules that are going to help me in actually carrying out that communication. That small signaling molecule is going to act as a messenger, basically, and pass on the message from one cell to the other. Now, when we talk about signaling molecules, so signaling molecules can be variety of different types, right? We can have different types of signaling molecule. We can, if we talk about it, we can have molecules that are hydrophobic. And if I were to give you an example of hydrophobic molecules, so hydrophobic molecules would be steroid. There are steroids. Your estrogen, your progesterone, your testosterone, your adrenal hormones. These are all steroid-based hormones. They're made up of lipids. They're steroid-based. So steroid, estrogen, progesterone, they're all steroid. So we can have hydrophobic molecules. The good thing about hydrophobic molecules is that they can diffuse directly to the cell membrane, right? They can diffuse directly through the cell membrane. And they can then go and directly diffuse to the cell membrane. And then, for example, they can go to the nucleus. And after going to the nucleus, they can tell the nucleus, listen, you need to start making this molecule. You need to start producing this compound. You need to start cell division, whatever the function of that product is. It will go and do that because it can directly cross the cell membrane. But more commonly, the more common type of signaling molecules that we see are water soluble. Now, this creates a small problem. How does this create a small problem? The problem it creates for us is this, that I just told you guys that the cell membrane is hydrophobic, right? It will only allow non-polar small molecules to cross through. So when a molecule is going to be water soluble, it cannot just directly diffuse to the cell membrane. So it actually needs to go through a signaling pathway, right? So what is going to generally happen? The general process is this, that the signal or the molecule will arrive to the protein receptor. Okay? The molecule will bind to the receptor on the cell membrane, right? So that's the first thing that it will have to do. It will have to bind to the receptor. After binding to the receptor, so the receptor will have a specific shape, and it will recognize this molecule. The receptors are shape specific. For example, if I am talking about your glucagon is not going to go and act on all the cells of the body. It's only going to act on those cells of the body that have the receptor to recognize. That's how the cells make sure that they are only only the cell that is the target is doing the job. I don't want, for example, if the message is to receive digestive enzymes, I don't want the message going to every single cell of the body. Release your digestive enzymes, right? Only the cells of the pancreas, only the digestion enzymes have to be released. So the messenger is going to be binding to specific receptors, right? That's how we make sure that only the ones that we want, only the tissue that we want is doing the job and not all the tissue in the body. So it will only recognize this particular molecules. That's it. Only the cells that have this receptor, only they will be able to understand the signal. The rest of them will not be able to. Now, what happens? The molecule brings about a change in the shape of the receptor. This molecule will change the shape of the receptor. And what happens? That since this receptor is span the length of the cell membrane. What do I mean by that? I mean, for example, this, the transmembrane molecule, right? It's going all the way through the cell membrane from one side to the other. So when we have molecules like these that span all the way through the length of the cell membrane, so what can happen as a result, right? What happens is that this signal is carried to the inside of the cell. We call this signal transduction. That's what we call it. That the signal has been carried on from the outside of the cell membrane to the inside of the cell membrane. Okay? So now the message has gone to the inside of the cell membrane. Is our story finished? Sadly, no. Our story continues. What happens after the message has gone to the inside of the cell membrane? Keep this in mind that it's still the inside of the cell membrane. It's still not all the way into the cell. It actually has to get all the way into the cell. How is it going to get all the way into the cell? The next component that we have is a certain protein. We can have different types of protein, by the way. This is the one that's in your syllabus, but we have different things can happen. So what I'm about to tell you, I'm going to tell you about G protein. This is only one type of pathway that can happen. When a signaling molecule binds to the receptor, different chemical reactions can happen. We are studying just one, and this is the most common one. So we are studying basically the most common methods, but there can be a lot of different types of chemical reactions that takes place. Now, what happens is often G protein, right? This protein acts as a switch. It basically acts like a switch. How does it act as a switch? It acts as a switch and it brings about the release of a chemical compound called a second messenger. So the signaling molecule was the primary messenger. We don't call it the primary messenger. That's just to explain, but it acts as sort of a primary messenger, bringing the message for the first time. That message is carried on from the outside of the cell membrane to the inside of the cell membrane. So inside of the cell membrane, we have the G protein. The G protein helps in carrying that message from the cell membrane to the second messenger. Now, the second messenger will then diffuse through the cell, relaying the message. Now, this second messenger is the one that's actually going to travel through the cell. It's going to go through the cytoplasm. It's going to go wherever it's needed. For example, it's needed in the nucleus. It wants to tell the cell to divide. So it will then go to the nucleus and it will pass on the message to the nucleus that, listen, you need to go ahead and divide, right? So this is how the system works. And if I go ahead and I show you, if I draw it for you, it looks like this. So we have, of course, we have our, if we talk about it, we are going to have our phospholipids, right? Now, among my phospholipids, let's say that I have two proteins. One protein that I have is the protein receptor. Okay. Now this is going to have some glycolipid component to it. Maybe. Okay. So what is the process actually going to be looking like? The process is going to be looking like this. This is my protein receptor. My signaling molecule is going to come and bind to the protein receptor. When it's going to bind to the protein receptor, what's going to happen? When it's going to bind to the protein receptor, let me write this down. This is the protein receptor. What's going to happen? This protein receptor will change its shape. And when it's going to change its shape, it's going to carry the signal to the inside of the cell to a very interesting protein that we call G protein. Right? Now, this G protein is going to carry on the message. In this particular instance, so this message can work in different ways. But in this particular instance, the message is being carried onto this enzyme. What does this enzyme do? This enzyme makes second messenger. The enzyme makes second messenger. So the enzyme is going to produce, when you're going to talk about it, it's going to go ahead and it's going to produce your second messenger. Now here is something very interesting. What is the interesting thing? The interesting thing is this, that one signaling molecule can lead to a lot of second messenger molecules being released. This one signaling molecule can cause the release of a lot of your second messenger molecules. So the binding signaling molecule is just one. But if you will take a look, you will see that one signaling molecule can cause the release of a huge number of second messenger. This is known as amplification. The signal, it's not just passing on from one to the other. The signal is not just passing on from the inside of the cell, sorry, outside of the cell to the inside of the cell. It's also getting amplified. It's also getting further stimulated. It's also getting further encouraged, right? So what actually ends up happening? What actually ends up happening is this: the one signaling molecule can cause the release of multiple second messengers. This leads to amplification of the signal. So not only is the signal passed on from the outside of the cell to the inside of the cell, the signal is also amplified. It's made much stronger, right? So this is one of the ways in which the cell can work. This is how your second messengers can work. So your signaling molecule or your G protein can work in different ways. Okay. The G protein can either activate an enzyme, like I explained to you guys. Over here, it was activating an enzyme, right? And the enzyme was producing our second messenger. That was one of the ways it works. Another way that it can go ahead and work is that it can do other things as well. I told you guys just one mechanism. We have different mechanisms. So when we talk about G protein is just one type. We can have cyclic AMPs. We can have different molecules. And then when we talk about G protein, one of the ways in which G proteins can produce second messengers is with the help of enzymes. Can also produce second messengers in other ways, right? For example, it can open an ion channel, right? It can tell an ion channel to open. So the ions can move from the inside of the cell to the outside of the cell, or from the outside of the cell to the inside of the cell, right? Or it can act directly as the membrane bound enzyme, or act directly as the membrane bound enzyme. So instead of producing the second messenger, the enzyme will act directly as an enzyme. Whatever the role of that enzyme is, it will do its job directly. That is another way that it can go ahead and actually do its job, right? So G proteins can either, activate the enzyme and tell the enzyme, listen, we need to start producing this molecule. We need to start producing second messengers. Or it can act on the enzyme and when you go ahead and talk about it, or when you talk about your signaling molecule. So signaling molecule, it's not necessary that the signaling molecule will act on the enzyme, right? Signaling molecule can act on the G protein, or the signaling molecule can open an ion channel, or the signaling molecule can act directly on the membrane bound enzyme, right? Whatever the enzyme is inside the membrane, so it will act directly as the membrane bound enzyme. Or it can act as an intracellular receptor. What is an intracellular receptor? So an intracellular receptor is a receptor that is going to bind to the molecule and then simply pass straight through the cell membrane. So no second messengers, nothing. Signaling molecule will bind to the intracellular receptor and pass straight through the cell membrane. So it can work in a variety of different ways. If you guys don't remember the different ways it can work, that's no problem. You don't have to go into details of it, but you do need to know the details of the G protein, how the G protein is specifically working. That you need to go ahead and know the concept of. You need to know the concept of what cell signaling is, how the G protein is involved in cell signaling, and then of course you need to know the rules of different components that are bound within the cell. Any confusion? Does anybody have any questions on the topic? Would anybody like to ask anything?
No.
Are you sure? All clear?
Yes.
Yes, ma'am.
All right. Okay, now after this, basically the last thing that's left in our topic of cell membrane is a topic that's quite small. I'm not expecting, it's something you guys have already an idea of. It's transport, movement of substances in and out of cell, right? So in movement of substances in and out of cell, you guys, I'm pretty sure, know really well diffusion, right? So we will actually, I'm sure you guys know the diffusion of the topic. We're just going to be taking a look at the factors that affect diffusion, and we're going to be seeing some really important examples within the body. Then, I have a type of diffusion that you guys have not studied before, that is facilitated diffusion. What is facilitated diffusion? Now, diffusion is the same. I'm moving from high concentration to low concentration, but I am facilitated, or I am helped by my proteins, by the transport channels. Remember, I said we would discuss them. The transport channels are helping me in the movement. So diffusion is when it happens by itself without help of anyone. Facilitated diffusion is still from high concentration to low concentration, but with the help of some transport molecules. Then, of course, we have osmosis, which again, I think you guys would already have a good idea of it. And then we have to cover active transport. Active transport, you guys would have already know the basics of it, but now we'll be going in a bit more detail in terms of how it actually happens. So we will be talking about carrier proteins. We will be talking about gated channels. We'll be talking about pumps. We have got pumps that actually help in pumping the molecules in and out of cell. And then of course we have to cover endocytosis and exocytosis. So that's the last thing that left in our topic, movement in and out of cell. So inshallah, we'll go ahead and take a look at that in the next class and inshallah finish this topic. Anybody has any questions? This is all clear?
Yes.
Okay. Okay, guys, then we will go ahead and stop it over here for today, and I'll see you guys in the next class. All right.